All right, jumping right into today's deep dive, we're going to be looking at cell signaling. Specifically, that fascinating overlap between JAK-STAT and RTK pathways.
Oh, those are two big ones.
Right. And, these are major players in how cells basically communicate, make decisions.
Absolutely.
And you've shared some interesting research with us. There were some excerpts about thyroid hormone receptors and cell signaling mechanisms. And basically, the mission here is you want to understand how these pathways intersect. Who are the key players? What does it all mean? What are the implications of these interactions for cell behavior? And I think the goal here is to really break it down step by step.
Okay.
Highlighting the why behind the what,
I like it. So what's fascinating is that while these pathways have their own distinct roles, they don't operate in isolation.
Right.
There's this crosstalk between them, a sort of cellular conversation that can significantly influence cell fate.
Okay, so kind of like how different departments in a company need to communicate to function smoothly, but within a single cell.
Exactly. Imagine JAK-STAT as the rapid response team to, say, an infection activating the immune system. Meanwhile, RTKs are more like the construction crews directing cell growth and development.
So we're not talking cell signaling 101 here, right? We're diving into how these two already complex systems intersect.
Absolutely. And a great example of this interplay is highlighted in the research you provided on thyroid hormone receptors.
Okay.
These receptors are part of the nuclear hormone receptor superfamily and are directly modulated by the thyroid hormone.
So that means these receptors are kind of like a bridge between the outside world where the hormone is and the cell's inner workings.
Precisely. Thyroid hormone binding directly impacts gene regulation and consequently influences a whole cascade of physiological processes. It really highlights how signaling pathways have a direct line to the cell's control center.
Okay, that makes sense.
Yeah.
But if both JAK-STAT and RTKs have such distinct roles, how do they actually, talk to each other? What's bridging the gap?
Great question. Adapter proteins are the key. Think of them as specialized communication hubs within the cell, facilitating the flow of information between JAK-STAT and RTKs.
So are they like translators converting signals between different cellular languages?
I love that. That's a great way to put it. They don't have intrinsic enzymatic activity, but their role as intermediaries is absolutely crucial. And what's really interesting, as highlighted in the research you shared, is that these interactions can be incredibly specific. Take the example of TREN1, a subtype of thyroid hormone receptor. It seems to play a distinct role in signal transduction, particularly during postnatal development.
Wow, so that's pretty amazing. It suggests that different adapter proteins might be involved depending on the stage of development. That adds another layer of complexity to the picture.
Absolutely. And that brings us to a crucial question. What happens when these pathways collaborate? Is it always a harmonious partnership, or can there be a conflict?
Well, right, because in any complex system, you'd expect some level of push and pull, right?
Exactly. The outcome of this crosstalk can vary depending on the context, what type of cell we're talking about, what other signals are present, and so on. Sometimes it leads to synergy, where the combined effect is greater than the sum of the parts, particularly in scenarios like cell proliferation and immunity.
Okay, so like a well-coordinated team effort.
Precisely. But in other situations, there might be competition for shared resources or even antagonistic effects. It's a delicate balance.
So let's talk about those STAT proteins. I remember from my reading that they're key players in the JAK-STAT pathway. They don't just sit around once activated, right?
Absolutely not. They have a crucial journey. Upon activation, they travel to the cell's control center, the nucleus, where they directly influence gene expression.
So they're carrying those important messages straight to the decision makers, huh? I'm thinking back to that source on cyclic AMP signaling. Wasn't there something about microdomains?
Yes. While still a bit controversial, the concept of microdomains is gaining traction. It suggests that even within the cytoplasm, signaling isn't always global. There can be localized compartments where specific signals are concentrated, adding another layer of complexity to the picture.
Okay, so it's not just about sending a message, it's about sending it to the right place at the right time. That's fascinating. But I have to ask, why should we care about all this molecular chatter? What's the relevance to us?
Well, it's crucial to understand because dysregulation in either JAK-STAT or RTK signaling can contribute to various diseases, including cancer and immune disorders. For example, your research mentioned resistance to thyroid hormone, RTH, a disease caused by mutations in the gene encoding TR, a type of thyroid hormone receptor.
So a single disruption in this communication pathway can have significant physiological consequences. That's pretty sobering.
It highlights how critical it is for these pathways to function correctly. but it also presents opportunities for developing targeted therapies.
Okay, I'm starting to see the bigger picture here. But before we get ahead of ourselves, can we break down some specific examples of this crosstalk between JAK-STAT and RTKs? You mentioned cytokine and growth factor synergy earlier. I'd love to hear more about that.
Absolutely. Cytokines and growth factors often work together to regulate cell behavior, and their signaling pathways, including JAK-STAT and RTKs, can intersect in various ways.
So can we dive into a specific example? It would really help to see how this intersection works in practice.
Let's consider interferon gamma, or IFNO, for short, an epidermal growth factor, or EGF. IFNO is a key player in the immune response, while EGF is known for stimulating cell growth and proliferation.
All right, so two very different functions, but both essential for the body. What happens when they both come into play?
When a cell is exposed to both IFNO and EGF, their respective receptors, which belong to the JAK-STAT and RTK families, are activated simultaneously.
So it's like receiving two important messages at the same time.
Exactly. And this dual activation leads to a fascinating interplay between the two pathways, often mediated by shared adapter proteins.
Those communication hubs we were talking about earlier.
Precisely. In this case, one of the key shared adapters is a protein called GERB2.
GERB2, okay. And how does it help facilitate this crosstalk?
GERB2 can bind to activated receptors from both the JAK-STAT and RTK pathways, essentially bridging the gap between them. It's like a dual-purpose bridge connecting two different communication routes.
So GERB2 is receiving signals from both IFNO and EGF receptors. What happens next?
When GERB2 binds to both types of receptors, it triggers a cascade of downstream signaling events that ultimately influence gene expression, particularly enhancing the expression of genes involved in both immune responses and cell proliferation.
So it's like a synergistic boost, amplifying the effects of both IFNO and EGF.
Precisely. This synergy is crucial because it allows cells to mount a more robust immune response while simultaneously promoting tissue repair and regeneration.
That's pretty incredible. So GERB2-mediated crosstalk is one way these pathways interact, but I'm guessing there are other ways they can influence each other.
Absolutely. Another fascinating example involves the direct phosphorylation of STAT proteins by RTKs.
Wait, so RTKs can directly activate STAT proteins even though they belong to a different pathway?
That's right. In some cases, activated RTKs can directly phosphorylate STAT proteins, bypassing the need for JAK kinases.
Wow, so it's like sending a very specific message directly to a particular department in the city, bypassing the usual communication channels.
Another perfect analogy. This direct phosphorylation mechanism adds another layer of complexity to the JAK-STAT RTK crosstalk, highlighting the versatility and adaptability of these signaling networks.
This is also fascinating, but it also seems incredibly complex. How do researchers even begin to untangle these interactions step by step? It's like trying to figure out the wiring of a supercomputer.
That's a great question. It is complex, and we're still uncovering all the intricacies, but there are some key steps researchers take to dissect these interactions.
Okay, so what's the first step?
First, they need to identify the specific receptors involved. Which JAK-STAT and RTK receptors are being activated by the specific stimuli being studied.
Right, so figuring out who's receiving the initial message.
Exactly. Then, they trace the downstream signaling events. This means mapping out the phosphorylation cascades, the adapter protein interactions, and the second messenger molecules that are generated downstream of receptor activation.
So following the trail of the message like a detective investigating a complex case.
Exactly. Next, they analyze gene expression changes. Which target genes have altered expression as a result of this crosstalk?
So figuring out what actions the cell is taking in response to the messages.
Precisely. And finally, they investigate the functional outcomes. What is the impact of this crosstalk on things like cell proliferation, differentiation, apoptosis, and immune responses?
Wow, so it's a systematic approach, starting from the initial signals and working down the chain to the ultimate cellular response.
Precisely. And it's a constantly evolving field, with new discoveries and insights emerging all the time.
Okay, that gives me a much better understanding of the research process.
Yeah.
But we've been talking a lot about synergy, especially between cell proliferation and immunity. Can you elaborate on that? I'm curious how these seemingly different processes are linked.
Absolutely. As we've discussed, the crosstalk between JAK-STAT and RTKs can lead to synergistic effects, where the combined effect of both pathways is greater than the sum of their individual contributions.
Right, like a well-coordinated team effort.
Yeah.
But how does this synergy work in practice?
Let's look at cell proliferation first. Often this synergy happens when growth factor signaling through RTKs enhances the effects of cytokines that signal through JAK-STAT.
So the growth factors are giving a boost to the cytokines.
Exactly. This boost can occur at various levels of the signaling pathways, ultimately increasing cell division and tissue growth.
Can you give me a specific example?
Consider the interplay between platelet-derived growth factor, PDGF, and interleukin-6, IL-6. PDGF is a potent stimulator of cell growth, and IL-6 is involved in inflammation and immune responses.
All right, so two important molecules with seemingly distinct roles.
Yes, but their pathways intersect. PDGF, signaling through its receptor, a member of the RTK family, can enhance IL-6 signaling through the JAK-STAT pathway. This leads to increased expression of genes that promote cell cycle progression, resulting in faster cell proliferation.
Wow, so it's like PDGF is giving IL-6 that extra push to accelerate cell division.
Precisely. Now, shifting to immunity, the synergy between JAK-STAT and RTKs often enhances immune responses, strengthening the body's defense mechanisms.
So they're working together to fight off invaders.
Exactly. And one way this happens is through the upregulation of immune-related genes and the modulation of immune cell activity.
Can you give me a specific example of this synergy in immunity?
Think about the interaction between interferon alpha, IFNA, and insulin-like growth factor 1, IGF-1. IFNA is a key antiviral cytokine, and IGF-1 is involved in growth and development.
Okay, so both important molecules, but again, seemingly with different roles.
Yes, but just like with PDGF and IL-6, their pathways can intersect. IFNI, signaling through the JAK-STAT pathway, can be enhanced by IGF-1 signaling through its receptor, which belongs to the RTK family.
So IGF-1 is giving IFNI a helping hand in fighting off viruses.
Precisely. This enhanced IFNI signaling leads to increased expression of genes involved in antiviral defense mechanisms, boosting the immune response against viral infections.
Wow, it's like the immune system's getting a power-up. So these examples really showcase the intricate ways these pathways collaborate, both for growth and for defense.
Absolutely. And it's just the tip of the iceberg when it comes to the complexity of JAK-STAT and RTK crosstalk.
I'm starting to get a sense of that. But all this talk about signaling pathways and molecular interactions is making my head spin a bit. Can we maybe revisit the concept of STAT translocation to the nucleus? I want to make sure I have a solid grasp on that process before we move on.
Of course. As we discussed, STAT proteins are key players in the JAK-STAT pathway, acting as messengers that relay signals from the cell surface to the nucleus.
Right, they're carrying those important messages to the decision makers.
Exactly. Now, when a cytokine or growth factor binds to its receptor, it triggers a cascade of events that ultimately lead to the activation of JAK kinases.
Those are the kinases specifically associated with the JAK-STAT pathway, right?
Yes. Once activated, JAK kinases phosphorylate specific tyrosine residues on the receptor. These phosphorylated tyrosine residues act as docking sites for STAT proteins.
So it's like the receptor is sending out a signal flare, attracting the STAT proteins.
A great way to visualize it. Once bound to the receptor, STAT proteins themselves are phosphorylated by JAK kinases.
So it's like the STAT proteins are getting a stamp of approval from the JAK kinases. But why is this phosphorylation so important?
This phosphorylation event is crucial because it causes the STAT proteins to dimerize.
Dimerize, meaning they pair up.
Precisely. The phosphorylated STAT proteins bind to each other, forming dimers. These dimers are then able to translocate to the nucleus.
So it's like they're getting their travel visas to enter the cell's control center. But how do they actually get inside?
The dimers, now carrying their NLS or nuclear localization signals, are recognized by important proteins, which act as chaperones, guiding them through the nuclear pores and into the nucleus.
So it's like a guided tour of the cell's control center. And once they're inside,
Once inside the nucleus, the STAT dimers bind to specific DNA sequences called STAT binding elements, which are located in the promoter regions of target genes.
so they're finding the right addresses within the cell's genetic blueprint.
Precisely. By binding to these promoter regions, the STAT dimers can either activate or repress the expression of the target genes.
So they're either turning the genes on or off, depending on the message they're carrying.
Exactly. This regulation of gene expression is how the JAK-STAT pathway ultimately influences various cellular processes.
Wow, it's like a complex chain of command, starting from the cell surface and culminating in the control of gene expression.
It truly is a remarkable system, highlighting the intricate coordination involved in cellular communication.
Okay, that was a great refresher on STAT translocation. But before we move on, I'm still curious about those STAT binding elements. You mentioned that there's specific DNA sequences, but what do they actually look like?
They're typically short stretches of DNA, usually around 10 to 20 base pairs in length.
So they're like tiny flags within the vast expanse of the cell's DNA.
Exactly. And different STAT proteins recognize and bind to slightly different STAT binding element sequences.
So it's like each STAT protein has its own preferred flag design. And this ensures that each STAT protein only regulates the expression of its designated target genes.
Precisely. This specificity is crucial for ensuring the accurate transmission of signals within the cell.
Okay, that makes sense. But how do STAT dimers actually activate or repress gene expression once they're bound to these promoter regions?
They recruit other proteins involved in regulating gene expression. Think of it as calling in reinforcements to help them carry out their mission.
Okay, so who are these reinforcements?
They can include coactivators, which enhance gene expression, or core pressors, which suppress gene expression.
So it's like having a team of builders to construct a building or a team of demolition experts to tear it down, all depending on the message the STAT dimers are carrying.
Another great analogy. And the specific coactivators or core pressors that are recruited depend on the specific STAT protein involved and the cellular context.
Okay, so it's a complex interplay of factors that ultimately determine whether a gene is turned on or off.
Precisely. And this intricate regulation of gene expression is what allows the JAK-STAT pathway to orchestrate such a wide range of cellular processes.
This is all incredibly fascinating. But you mentioned earlier that both JAK-STAT and RTK pathways can lead to complex outcomes. And I'm curious to explore that further. Can you elaborate on how the interplay between these pathways contributes to this complexity?
Absolutely. As we've discussed, both JAK-STAT and RTK pathways can be activated by a variety of ligands and can influence a wide range of cellular processes.
Right, it's not just a simple on or off switch.
Exactly. And this complexity is further amplified when these pathways intersect, leading to crosstalk.
So the crosstalk adds another layer of intricacy to the mix.
Precisely. The outcome of this crosstalk can vary depending on a multitude of factors.
Can you give me some examples of these factors?
Of course, the specific ligands involved are crucial. Different ligands can activate different combinations of JAK-STAT and RTK receptors, leading to distinct downstream signaling events.
Okay, so the initial message can shape the entire course of the conversation.
Exactly. And then there's the cellular context. The same ligand can elicit different responses in different cell types, or even in the same cell type under different conditions.
So the environment in which the conversation is taking place also matters.
Precisely. And finally, the presence of other signaling pathways adds another layer of complexity. The JAK-STAT and RTK pathways don't operate in isolation. They can interact with other signaling pathways within the cell, creating a complex network of interactions.
So it's like having multiple conversations happening simultaneously, all influencing each other.
That's a great way to visualize it. Now, this complexity can lead to outcomes that are sometimes difficult to predict.
Can you give me an example of this unpredictability?
Sure. Consider the role of JAK-STAT and RTK crosstalk in cancer development. Both pathways have been implicated in cancer, but the outcome of their interaction can vary.
So sometimes this crosstalk can promote tumor growth, and sometimes it can suppress it.
Exactly. In some cases, the crosstalk between these pathways can promote tumor growth. For instance, in some cancers, growth factor signaling through RTKs can activate STAT3, a key player in the JAK-STAT pathway that promotes cell survival and proliferation.
So in that scenario, the communication between these pathways is essentially driving the uncontrolled growth of the cancer cells.
Precisely. But in other contexts, the crosstalk between JAK-STAT and RTKs can actually have anti-tumor effects. For example, in certain cancers, IFNS signaling through the JAK-STAT pathway can inhibit tumor growth, and this effect can be enhanced by growth factor signaling through RTKs.
Wow, so in that case, the growth factors are actually helping to suppress the tumor.
Exactly. This highlights the complexity and context-dependent nature of JAK-STAT RTK crosstalk, demonstrating how the same pathways can have both positive and negative effects depending on the specific circumstances.
This is all incredibly intricate, but I'm starting to get a better grasp of how these pathways work together and how their interactions can lead to such diverse outcomes.
That's great to hear. It's a testament to your curiosity and your willingness to delve into the complexities of cellular communication.
Now, I know we've covered a lot of ground, but I'd love to circle back to STAT translocation to the nucleus. You mentioned earlier that this translocation typically occurs after STAT proteins have been activated by phosphorylation. Can you elaborate on when this translocation actually happens?
Of course. As we discussed, STAT translocation to the nucleus is a crucial step in the JAK-STAT signaling pathway. It typically occurs after STAT proteins have been activated by a phosphorylation.
So it's like they need that stamp of approval before they can enter the cell's control center.
Precisely. The phosphorylation event triggers a conformational change in the STAT proteins, exposing a nuclear localization signal, NLS.
A nuclear localization signal, what's that?
It's essentially a molecular tag that acts like a passport, allowing proteins to enter the nucleus.
So it's like the STAT proteins are getting their entry visas.
Exactly. The NLS on the activated STAT proteins is recognized by important proteins which act as chaperones, guiding the STAT proteins through the nuclear pores and into the nucleus.
So it's like a guided tour of the cell's control center.
Yeah.
And what happens once they're inside?
Once inside the nucleus, the STAT proteins can carry out their mission of regulating gene expression.
Wow, it's amazing how all these molecular events are coordinated to ensure that the right signals are delivered to the right places at the right times.
It truly is a marvel of cellular communication, and the timing of STAT translocation can vary depending on the specific STAT protein involved and the cellular context.
So there's no fixed schedule for these molecular messengers.
Not exactly. Some STAT proteins translocate to the nucleus very rapidly after receptor activation, while others take a bit longer.
I'm guessing this difference in timing is related to their specific functions.
You're absolutely right. The timing of STAT translocation is often fine-tuned to ensure an appropriate cellular response to a particular stimulus.
Okay, so it's like a well-choreographed dance where each STAT protein enters the stage at precisely the right moment.
A perfect analogy. Now, to delve a bit deeper into where these cascades converge, we need to understand that both JAK-STAT and RTK pathways eventually converge on the regulation of gene expression.
Okay, so they ultimately meet in the cell's control center, the nucleus.
Precisely. While they take different routes to get there, their ultimate goal is to modulate the activity of specific genes.
So it's like two rivers flowing into the same lake.
Exactly. And this convergence on gene expression regulation can manifest in various ways.
Can you give me some examples of how this convergence occurs?
Of course. One way is through the direct binding of STAT proteins to STAT binding elements in the promoter regions of target genes.
We talked about that earlier, right?
Yes. This direct binding can either activate or repress gene expression, depending on the specific STAT protein involved.
Okay, so that's one point of convergence. What are some others?
Another way is through the modulation of other transcription factors by the JAK-STAT and RTK pathways.
Transcription factors. Those are proteins that control gene expression, right?
Exactly. The JAK-STAT and RTK pathways can activate or inhibit other transcription factors, indirectly influencing the expression of their target genes.
So it's like a chain reaction where one pathway influences another, ultimately leading to changes in gene expression.
Precisely. Now, this convergence on gene expression regulation can have profound effects on cellular processes.
Can you give me some examples of these effects?
Sure. For instance, the crosstalk between JAK-STAT and RTKs can influence cell proliferation, differentiation, apoptosis, and immune responses.
So it's like a master control panel regulating a wide range of cellular activities.
Exactly. And the specific outcomes depend on the intricate interplay between these pathways and the cellular context.
This is also fascinating, but I'm curious, how do researchers even begin to tease apart these complex interactions? It seems like a daunting task.
It definitely can be challenging, but researchers use a variety of techniques to unravel the intricacies of JAK-STAT-RTK crosstalk.
Can you walk me through some of these techniques?
Of course. One common approach is to use specific inhibitors to block the activity of certain proteins involved in these pathways.
So it's like shutting down specific communication channels to see how the system responds.
Exactly. By selectively inhibiting different components of the JAK-STAT and RTK pathways, researchers can determine the contribution of each pathway to a particular cellular response.
Okay, so that helps to dissect the roles of each pathway. What are some other techniques?
Another approach is to use genetic manipulation techniques, such as gene knockout or gene overexpression, to alter the expression levels of key proteins.
So it's like tinkering with the cell's genetic blueprint to see how the changes affect the communication network.
Precisely. By manipulating gene expression, researchers can study the effects of specific proteins on the crosstalk between JAK-STAT and RTKs.
Okay, so that provides insights into the functions of individual proteins. Are there any other techniques?
Yes. Researchers also use biochemical assays to directly measure the activity of specific proteins, such as kinases and transcription factors.
So it's like taking a closer look at the molecular machinery to see how efficiently it's working.
Exactly. These biochemical assays provide quantitative data on the activity of key proteins, allowing researchers to assess the impact of various stimuli or inhibitors on the JAK-STAT and RTK pathways.
Wow, it's impressive how researchers combine these different techniques to piece together the puzzle of JAK-STAT RTK crosstalk.
It's a testament to their ingenuity and perseverance in unraveling the complexities of cellular communication. It truly is remarkable how these pathways have evolved to ensure cells can adapt and thrive.
It really makes you appreciate the incredible complexity happening within us, right down to the tiniest levels.
Absolutely. And speaking of intricate details, we've mentioned adapter proteins quite a bit. They seem to be the key connectors in this cellular communication network. Could you tell me more about their specific roles in JAK-STAT and RTK crosstalk?
Yeah, I'm especially interested in how they contribute to that synergy we were discussing earlier between things like cell proliferation and immunity.
That's a great point. Adapter proteins are fascinating because they act as bridges, linking different components of these signaling pathways together.
So they're like the linchpins that allow the different parts of the system to communicate effectively.
Exactly. They facilitate interactions between receptors, kinases, and other signaling molecules involved in both JAK-STAT and RTK pathways.
Do you have any specific examples of these adapter proteins in action? I'd love to see how they actually work within these pathways.
Of course. We've already touched on group 2, which plays a key role in connecting both JAK-STAT and RTK receptors.
Right, that dual-purpose bridge that can bind to both types of receptors.
Precisely. Group 2 has these special regions called SH2 and SH3 domains, which allow it to bind to specific phosphorylated tyrosine residues on activated receptors.
So those domains are like hooks that latch onto the receptors.
Exactly. And once GRO2 is bound, it can recruit other signaling molecules like SOS, which is a ROS-guanine nucleotide exchange factor. This ultimately leads to the activation of downstream pathways like the MAP kinase pathway.
Okay, so GRB2 is like a central hub, connecting JAK-STAT and RTKs to other signaling networks within the cell. It's starting to feel like a very complex web.
It is. And another important adapter protein is AC, which can also bind to both activated RTKs and JAK-STAT receptors.
So SureC is another bridge builder, helping to facilitate communication between these pathways.
Absolutely. SureZ uses a slightly different mechanism, relying on its phosphotyrosine binding PTB domain to attach to phosphorylated tyrosine residues on the receptor.
So a different kind of hook, but with a similar outcome connecting the pathways.
Exactly. Once SureC is bound, it can be phosphorylated by receptor tyrosine kinases, and this creates binding sites for group 2, so you can see how they often work together to amplify the signals coming from activated receptors.
It's amazing how all these pieces fit together, but these examples seem to focus on connecting receptors to downstream signaling molecules. Can adapter proteins also connect different parts of the JAK-STAT and RTK pathways to each other?
That's a great question, and the answer is yes. A good example is the adapter protein GAB2, which has been shown to play a role in the crosstalk between the IL-6 receptor, a member of the JAK-STAT family, and the EGFR, a receptor tyrosine kinase.
So GAB2 is like a specialist, bridging the gap between those two specific receptors.
Precisely. GAB2 has multiple tyrosine residues that can be phosphorylated by JAK kinases activated by the IL-6 receptor.
So GAB2 is getting that stamp of approval from the JAK kinases.
Exactly. These phosphorylated tyrosine residues on GAB2 then become docking sites for proteins containing SH2 domains, including the P85 subunit of PI3 kinase, a key enzyme in the RTK pathway.
Okay, so GAB2 is bringing PI3 kinase into the picture, essentially connecting the IL-6 signal to the RTK pathway.
Exactly. This recruitment of PI3 kinase to the IL-6 receptor complex then activates downstream signaling events typically associated with the RTK pathway.
It's fascinating how adapter proteins can blur the lines between different signaling pathways.
They really do. This example with GAB2 shows how adapter proteins can enable pathways to share components and influence each other's activities in complex ways.
I'm starting to see adapter proteins as the master conductors of this cellular orchestra, coordinating all these different signals and ensuring that the right messages get to the right places.
They're essential for maintaining the smooth flow of information within the cell.
You mentioned earlier that researchers face some significant challenges when trying to study these interactions. I can only imagine how difficult it must be to tease apart all these intricate details.
It's definitely a challenge, but a fascinating one. One of the biggest hurdles is the sheer complexity of these signaling networks. We're dealing with multiple pathways, countless proteins, and an incredible number of interactions.
It's like trying to untangle a giant ball of yarn, but with molecular-sized threads.
That's a great analogy. Another challenge is that these pathways are incredibly dynamic. Signaling events are constantly changing, making it difficult to get a clear picture of what's happening at any given moment.
So it's like trying to hit a moving target, but at the microscopic level.
Exactly. And to make things even more complex, our current research tools have limitations. While we have techniques to study individual components, it's still very difficult to fully grasp the interplay between multiple pathways in real time.
So it's like trying to understand how a complex machine works by only looking at individual parts without seeing how they all fit together.
Precisely. But despite these challenges, researchers are making significant progress in unraveling the intricacies of JAK-STAT and RTK crosstalk.
That's really encouraging to hear. Are there any particularly promising avenues of research in this field?
One exciting area is the development of new imaging techniques that allow us to visualize signaling events in living cells.
So we can actually see these interactions happening in real time? That's amazing.
It is. These new imaging techniques are providing unprecedented insights into the dynamics of signaling pathways. Another promising area is the development of computational models that can simulate signaling events.
So it's like creating a virtual cell where researchers can experiment with different scenarios.
Exactly. These models are helping researchers test hypotheses and predict the outcomes of different perturbations to the signaling network.
Wow. It's like having a crystal ball that can predict the future of cell signaling.
In a way, yes. These computational models are becoming increasingly sophisticated, allowing researchers to make more accurate predictions about the behavior of these complex pathways.
This is all incredibly exciting. But I'm also curious about the practical implications of this research. How can understanding JAK-STAT and RTK crosstalk lead to new treatments for diseases?
That's a crucial question. As we discussed, when these pathways are dysregulated, it can contribute to a wide range of diseases, including cancer, autoimmune disorders, and inflammatory diseases.
Right, so by understanding how these pathways go wrong, we can potentially develop strategies to correct them.
Exactly. One promising approach is to develop drugs that target specific components of the JAK-STAT and RTK pathways. Think of it like having a toolkit that allows us to fine-tune the cell's communication system.
So we can potentially adjust specific signals or interactions to restore balance and treat the disease.
Precisely. For example, JAK inhibitors are already being used to treat certain types of cancer and inflammatory diseases.
That's amazing. A real-world example of how this research is translating into tangible benefits.
Yes, and there are many more potential targets within these pathways that are being explored. Researchers are looking into inhibitors of specific adapter proteins, as well as drugs that target downstream signaling molecules like MAP kinases and PI3 kinase.
So it's a multi-pronged approach, targeting different parts of the communication network to achieve the desired outcome.
Exactly. And one of the most exciting aspects of this approach is the potential for personalized medicine, tailoring treatments to the specific molecular profile of a patient's disease.
So it's not a one-size-fits-all approach anymore. It's about understanding the unique communication breakdowns in each patient's cells and finding the right tools to address them.
Precisely. As our understanding of JAK-STAT and RTK crosstalk deepens, we can expect even more targeted and effective treatments to emerge.
Exactly. For example, certain cytokines like TNF-alpha can activate the JAK-STAT pathway, leading to the expression of pro-apoptotic genes, which then trigger cell death.
So TNF-alpha is like a messenger of doom, delivering the signal for the cell to self-destruct. Yes, and this signal can be amplified by growth factor withdrawal or the activation of certain RTKs that promote apoptosis.
Okay, so it's like a double whammy, with both pathways contributing to the cell's demise.
Precisely. But on the other hand, growth factor signaling through RTKs can also activate survival pathways that inhibit apoptosis.
Well, it's like those growth factors are throwing the cell a lifeline, preventing them from self-destructing.
Exactly. And these survival pathways often involve the activation of PI3 kinase and ACT, which suppress the activity of pro-apoptotic proteins.
It's like a constant tug-of-war between pro-apoptotic and anti-apoptotic signals, and the balance between JAK-STAT and RTK pathways can tip the scales in either direction.
Precisely. And this delicate balance is crucial for maintaining healthy tissues and preventing diseases like cancer, where uncontrolled cell survival can lead to tumor growth.
This is all so intricate. It's mind-boggling how these pathways are involved in so many critical cellular decisions.
It really speaks to the complexity and elegance of cellular communication. Now, let's move on to our final cellular process, immune responses. Okay, the body's defense system against those invaders.
Exactly. And the interplay between JAK-STAT and RTK pathways is absolutely essential for coordinating a robust and effective immune response.
I'm ready for more insights. How do these pathways actually contribute to our immunity?
One way is through the activation of immune cells like T cells and B cells, which are the key players in adaptive immunity.
Adaptive immunity, that's the part of the immune system that learns and remembers specific pathogens, right?
Oh, precisely. And the activation of T cells and B cells often requires signals from both cytokines acting through the JAK-STAT pathway and antigens, which are molecules derived from those pathogens.
So the cytokines are like the alarm bells, alerting the immune system to an intruder, while the antigens are like the wanted posters, providing specific information about the enemy.
That's a great analogy. Cytokine signaling through the JAK-STAT pathway often provides the initial signal to activate T cells and B cells, promoting their proliferation and differentiation into effector cells.
Effector cells, those are the ones that actually carry out the attack against the pathogens, right? Exactly. For example, helper T cells activated by cytokines like IL-2 can differentiate into effector cells that secrete cytokines to stimulate other immune cells, while cytotoxic T cells activated by cytokines like IF and gamma can directly kill infected cells.
So the JAK-STAT pathway is mobilizing the troops and getting them ready for battle.
Precisely. And antigen signaling, often through RTKs, provides the specificity to the immune response, making sure the immune system is targeting the right enemy.
So it's like the RTKs are providing the target coordinates for the immune cells to zero in on.
Exactly. For instance, B cells activated by both cytokines and antigens can differentiate into plasma cells that produce antibodies, which are highly specific proteins that bind to and neutralize pathogens.
It's a coordinated effort, with JAK-STAT providing the activation signal and RTKs providing the target specificity.
Precisely. And this cooperation between the pathways is essential for mounting an effective and targeted immune response. It's a beautiful example of how cells use multiple layers of communication to protect the organism.
This has been an incredible journey through the world of cell signaling. We started with the basics and ended up exploring the intricate dance between JAK-STAT and RTK pathways, uncovering the key players, the points of convergence, the mechanisms of synergy, the importance of cellular context, and the implications for so many essential cellular processes.
And we've only just scratched the surface. There's so much more to discover about this fascinating field.
It's truly mind-blowing to think about all this complex communication happening within us at the microscopic level. It's a testament to the incredible power and adaptability of life.
I couldn't agree more. As we continue to explore the intricacies of these pathways, we're bound to uncover even more amazing insights into the workings of life itself.
Thank you so much for being our guide on this deep dive. I feel like I've gained a whole new appreciation for the complexity and beauty of cellular communication.
It's been my pleasure. Your thoughtful questions and enthusiasm have made this a truly rewarding conversation.
I can't wait to see what new discoveries await us in this exciting field.